In this video, I'll be demonstrating meiosis. Meiosis is the type of division that happens only in reproductive cells. So it's the type of division that will give rise to the gametes, the male and the female gametes, that is the sperm and the eggs. As with the other types of cell division, we are going to start here. This is in eukaryotic cells, which are usually diploid. Diploid meaning that they have two sets of chromosomes. So we have two sets of chromosome number 1, and we have two chromosomes for two versions of chromosome number 2. So these two will be homologous chromosomes. These are the homologs for chromosome number 1, and this will be homologous. So this is the homolog for chromosome number 2, and so on. Before we can do any type of cell division, the first thing will be to replicate the DNA. So we did the same thing in mitosis, and we're doing that again here in meiosis. So again, during the synthesis phase of the cell cycle, the cell will make as identical as possible copy of its DNA. So all the chromosomes will get replicated, and they will have exactly the same information. So the same allele that is present here will be present on this chromosome. So I already have those made. The first thing that we'll have is replicate the DNA. We have same alleles, and the replicate will remain attached, and it's called a sister chromatid. This is a repeat from last video. Now we have the same 4 chromosomes, because as long as the sister chromatids are attached, they're still called chromosomes, so there's still 4 chromosomes. And these two are still the homologous chromosomes, and these two are still the homologous chromosomes. So remember, even if this has 2 sister chromatids, it's still called a single chromosome. Okay, so now we're ready to start meiosis 1. The first step in meiosis 1 is prophase, prophase 1. And in prophase 1, the chromosome condense and become visible, like we have them here. And an unusual thing that only happens in meiosis 1, the homologous chromosomes now come together. So these two come together, and these two come together. And this is called synapse. They come together and they start associating with each other. And there is something else that happens here, and is that they start exchanging pieces of information. So a piece of this chromosome will be swapped with the other chromosome. So for example here, if this piece gets swapped with the other chromosome, with its homologous, now this guy will take the gene for red circles, and the other chromosome now has the gene for white circles. And let's say the same thing happens here, and they swap this one of these. It doesn't have to be the middle one if 2 chunks switch back and forth. We have this exchanging different pieces of information. And this is called crossing over. So this is the process of crossing over that is very important in providing genetic variation. So these chromosomes originally in the cell that is doing cell division, that is about to produce gametes, one of these chromosomes came from one parent and the other chromosome came from the other parent. So this will be the baby's grandparents. And if there was no crossing over, the chromosomes will remain just as this cell received them from this cell's parents. But thanks to crossing over, there is an exchange, and let's say between grandma and grandpa's information. So we have crossing over happening in prophase 1. Now, as we move to the next step in meiosis, the chromosomes will align on the metaphase plate. We can already have them here, but they will move around and they will migrate. An important thing comparing mitosis and meiosis is that in meiosis 1, the spindle fibers will attach not to both sides of one chromosome, but only to one of the chromosomes. So they will attach to this chromosome, and then they will attach to this other chromosome. Okay? And these chromosomes at this point are connected by their synapses. Same thing will happen here. So these two are connected by their synapses, and the spindle fibers are just attached to one chromosome on each side. In Anaphase, the proteins that are connecting the homologous chromosomes together will break down. So now the homologous will separate, and they will migrate. They're pulled by the spindle fibers, and they will migrate to the different ends of the cell. So this will be anaphase, and finishing here with telophase. And then we have cytokinesis, where we split these chromosomes. Split those cells, divide the cytoplasm of the cells. So now we have 2 cells. But notice that now each of those cells only has 1 set of chromosomes. For chromosome 1, we only have 1. And for chromosome 2, we only have 1, which we used to have 2 of each. So now the homologous have separated. These cells are now haploid. They only have 1 set of chromosomes. However, because of crossing over, the sister chromatids are no longer identical. So they exchange pieces with the other homolog, and now they have recombinant information. So they're mixed between the 2 original homologous chromosomes. So this will be the end of meiosis I, and now the cell will do the second set of division, which is meiosis II. In meiosis II is very similar to mitosis. Here, what happened in meiosis II is the chromosomes in both cells will migrate to the center, which we already have them, in the metaphase plate. And just as we had in mitosis, the centromeres, the proteins that are keeping those sister chromatids together, will break down. So this will be anaphase in meiosis II. Anaphase II will separate the sister chromatids. So now they're separate, and the spindle fibers that are pushing those chromosomes apart will separate them. So that will be telophase. And then we'll have the new cytokinesis. Same thing will happen in this cell. Now this is just for illustration purposes, but they will be completely separate. There will be 4 independent cells, each with just 2 chromosomes. So we started with a cell that had 4 chromosomes, 2 pairs. Now we have 4 cells that have only 1 set of chromosomes. So 1 for chromosome 1, 1 for chromosome 2. And if you look around, every cell is different from the other. They all have completely different genetic information. So this one has white circles. This one has white circles with red triangles. This one has red circles with red triangle, and this one red circles with white triangles, and white circles and white triangles. So every single cell here has very different information. So if this was, for example, seed color, this will have yellow seeds, and this one has green seeds, but this one has one type of flower color, and this one has a different type of flower color. If this is purple flowers, this both will have purple flowers, but here we'll have white flowers, and so on. So now all the information in these cells is completely different. So these are the ways in which meiosis contributes to genetic variation.